Okay, I'm going to go ahead and start. Hi all, thank you for joining us. This is the CaTS COVID-19 Chronicle Seminar Series. We are proud of our scientific response to the COVID-19 pandemic. Researchers from around the state are working on the front lines and conducting research to inform testing and surveillance, health impacts, and treatment. As a resource to all scientists, clinicians, and citizens, we have launched the Louisiana COVID-19 Chronicle Seminar Series beginning today and then continuing every other week. Our first session today is hosted by Dr. Shefflin, MD at Tulane University. Dr. Shefflin?
Great. Thank you very much, and thank everybody for joining in today. I think we have a good lineup of speakers. So to start us off is Dr. Lucio Miele, who is a professor and department head at the Department of Genetics at the LSU School of Medicine in New Orleans. He's also the director of interinstitutional programs at the LSU Stanley Scott Cancer Center and the Louisiana Cancer Research Consortium, and the Cancer Crusaders Endowed Professor of Cancer Research. He is also the director of the CaTS Biomedical Informatics Core. So joining him in his presentation is Dr. Ron Horswell, who is an associate professor at Pennington Biomedical Research Center, and he also serves as the Data Access Group Director of the CaTS Biomedical Informatics Core. So Dr. Miele, please start off and take us away.
Thank you so much. As you can see from the first slide, hang on, I'm going to share content. Okay, you should all be able to see this slide now. As you can see, in addition to Ron and myself, members of our team include Dan Fort, who is the head of data analytics at Oxner, San Chu, who is also part of the Pennington contingent of the Biomedical Informatics Core, and possibly the most essential member of the team, as well as Chindo Hicks, who is a professor of bioinformatics and genetics here at LSU. So from the standpoint of biomedical informatics, the most important update that I have for the group is that of our participation in N3C. N3C is the national collaborative COVID-19 cohort. This is an effort funded by NIH, specifically by NCATS, the National Center for Advancement of Translational Science, NIGMS, which is the institute that funds the CaTS grant, joined in with a supplement. The supplement involves 7 different idea CTRs, including CaTS. The lead institution is the University of West Virginia. So together, the 7 of us have received a supplement to contribute data to the N3C. The data contributing organizations are LSU Health Sciences Center /UMC, Oxner, and Tulane /HCA. However, all members of CaTS institutions can apply to access the data either individually or by joining existing work groups and perform research on this data set. As we'll see in a second, I'm going to go through some detail. This is a centralized data repository. It's a secure analysis space where you essentially upload your question, create a Jupyter notebook, and perform your analytics behind the NIH's firewall, similar to what happens with the All of Us research program. There's no downloading of data allowed. This is from a presentation that NIGMS and NCATS gave us. Now, what this N3C is, it's an aggregate of clinical lab and, if, when possible, imaging data from health provider organizations. These are updated on a weekly basis, so they are longitudinal data. Therefore, they will allow to follow participants in time. And eventually also identify patients who might wish to participate in clinical trials. NCATS put this together very quickly, taking advantage of the experience accumulated with all of us. The CDC, the Food and Drug Administration, and a number of other organizations are also involved in the management of this cohort. As I said earlier, there is a secure data enclave that investigators can access, and I'll tell you in a second how exactly that is accomplished. This is not a federated database. We can, and we've discussed the potential to create a federated database within the CTRs, because our patient populations have unique characteristics. This is a centralized database. There are pros and cons with each of them. The reason why NIH elected to make it centralized is so as to apply machine learning and artificial intelligence in general to analytics of this data set. The data formats that are allowed, this is actually a very nice feature of this program. It imports data in whatever format we have them. ACT is the data ontology of the of NCATS. Trinetics is an industry-backed data ontology engine that's built to identify potential participants in clinical trials. PCORI Common Data Model is the data ontology used by PCORNet, and OMAP, Observational Medical Outcomes project is probably the most complete of these data sets. So you can contribute data whichever way you want. They are harmonized within the system, and they are now stored in this large data set within the NCATS firewall. What we're doing is contributing data in OMAP format for LSU UMC and PCORI CDM format for Tulane. I believe also for Oxner, but I may be wrong. These are the websites of the organizations. Particularly the onboarding one is important because it will allow you to have access to the data, which is not necessarily a straightforward process. Can you see this? Registration and onboarding?
Yes, we can.
All right. This is where you click to register. We're not going to go through the whole process. We're going to go back to the slides, but I just wanted you to see this. Now, there are 3 levels of data within N3C, and the level of clearance required to access them is different. They have synthetic data. Now, these are truly synthetic. I have confirmed that with NCATS. In other words, they are not scrambled. They are completely artificial data. And you don't even need to have city training to access this data. You do need to be from an organization that has a data user agreement with NCATS, and we'll talk about that process in a second. Then you've got de-identified data, which include data stripped of the 18 HIPAA identifiers. And then you have a limited data set, which includes also zip code, which you're going to use if you're going to need to do geocoding type studies. Now, this is what you need. You need to click on that link that I showed you earlier to register as a user. You can only register if your organization has a executed data user agreement with NCATS. You don't sign the DUA as an individual investigator, your organization does. For LSU Wellsett Science Center, I've taken it upon myself to route this. Each organization needs to do its own, and a number of them have already been executed within LCATS. Then the investigator signs in to become a data user. Unfortunately, you have to go through some data security training that is specific for this site on the NIH's website. And then you can submit your data user request. Your data user request is rapidly, I mean, this looks like it's a lot of work. This is all done very quickly by the Data Access Committee, and then you're allowed into the enclave to do your research. This is the form for the DUA. I'm not going to pull it for the sake of time. However, I will tell you that the language is non-negotiable. So when if you're part of a LCATS organization that hasn't executed one of these, it's important that you communicate this to your administrative leaders that this DUA cannot be modified. What is allowed? COVID-related research only, no re-identification of individuals, no downloading, as I told you earlier, and the platform is open to all researchers. All activity on the enclave are recorded and can be audited. And where analytics come from, you can upload analytical script, but where exactly it's coming from is being recorded. So once you are registered, you have to have an ORCID ID. This is another important piece of information. If you're an investigator who wants to use this data set, you have to have an ORCID ID. The process of obtaining one is very simple, and these are used to track all your academic activities online. I've had one for years. You have to show that you have city training if you're going to seek access to either de-identified or limited data set data. And you have to complete these training programs on the NIH's website. If and only if you're requesting access to a limited data set, you also need your own IRB to approve the study. And you need to upload a PDF of your IRB approval. If you're accessing the synthetic data or the fully de-identified data, you do not need that. Then you do your own data user request, the DUR. This is how it works. Title of my investigation, say effect of XYZ on the outcome of mild to moderate COVID-19. You have to explain the rationale. And as I said, if applicable, you upload your IRB approval. The Data Access Committee, now I've spoken with folks from the Data Access Committee. Essentially, their job is to determine what level of data you need. They're always going to give you something. So they emphasize this. The point is not to block you from accessing the data. If you can demonstrate you truly need the limited data set, you'll get that. If your work can be done without using the limited data set, you'll still get access to the de-identified data. You're not going to be bounced back and have to resubmit. And then you enter the workbench. This is your login screen, which you can log in with your ERA Common ID. That's probably the easiest one. There's a two-factor authentication, and this is what you see in the data enclave. Now, you can do two things. Either create a project, but you can also join an existing project. And there's a screen where you can see the projects that are already ongoing. This is very team science oriented. You'll have an actual number of patients in the repository up to the point where you're, this is a static screenshot. So up to the point where you're logging in, how many sites, drug exposures, procedures, etc., etc. This is how you enter, you create a project. You enter your project ID. Again, do you allow our researchers to join or not? So you have an option to do so or not. Non-confidential statement. And this is what level of data you require for what reason. This is how you justify what tier of data you need. Honestly, my opinion is that the truly synthetic data are useful for teaching purposes and also to check your script if you've come up with an analytical algorithm that you want to make sure it works. But you're not going to make discoveries off of the synthetic data set. You're actually going to need either the identified or the limited data set. And this is how you get you submit, and then you get your determination. This is the decision-making process for the data access committee. If it's not COVID, reject. If it's COVID, yes. What tier is requested? If it's limited data set, do these investigators really need zip codes and dates? If yes, we approve. If no, de-identified, yes. Are the 18 identifiers required? If so, yes, and approve. If no, approve, but provide access to the limited data, to the de-identified data set. Now, this is how you enter a project as a collaborator, and you have to attest that you completed all the data information security and human subjects research training, and agree to abide by the N3C code of conduct. This is the enclave. You can see the existing projects, and if you're interested in something and you want to join, you can request to join. This is your personal homepage within the enclave. Now, done with N3C. There are a number of other projects that the Biomedical Informatics Group has either started or enabled prior to our entry into N3C. We have an umbrella-wide data project protocol under which we can file sub-projects. There are 4 that are ongoing right now. One on risk factors, predicting critical illness in hospitalized patients, estimating probability of ventilator use, and COVID testing at LSU HSC. I know this is our own internal testing program. You've got the oxner seroprevalence or seropositivity studies, on which we have 2 slides in a little bit. The LSU campus asymptomatic case survey. This is something that we have been doing for the past 3.5 months. We're surveying both antibody and virus testing in asymptomatic students, staff, and faculty on the New Orleans campus. There is a biorepository that's primarily from early disease. It's based at LSU Health Sciences Center in New Orleans, and it includes blood, plasma, urine, stool, and nasopharyngeal swabs, as well as data. The data are going to be in the same format as we upload to N3C, so Common Data Model or OMAP. And essentially, these biospecimens, of which we already have a few dozen, are linked to their relevant clinical data. There may be a separate biorepository at Pennington, depending on whether or not an LSU-wide screening program and statistical survey of virus positivity is funded. That's now all tangled with what's going on in Congress with the second stimulus package. And we have a pending GRADEX application for next generation sequencing-based mass testing and virus sequencing in New Orleans. This slide was produced by Ron, and it's very informative in giving you a snapshot of the variables that can be studied at each stage. So in the general population, of course, you can study behavioral and biological factors affecting risk, as well as public health and control strategies. From the biological standpoint, but innate immunity, vaccine development and administration. Between incidence and progression, you can study either the factors affecting initial progression or early treatment strategies. And we have studies on both. I'm going to have to switch screens that I share. These are the studies that we have ongoing on factors affecting initial progression. They have to do with diabetes, clinical features in general, BMI, bitter taste receptor, race disparities, and again, obesity, and more obesity. For early treatment strategy, we have these 3 studies going on. Factor 10A, anti-COVID-19 therapeutics. This is a broad study, and thrombin inhibition. We actually opened another one here at LSU New Orleans, which is the Eli Lilly spike monoclonal antibody active 3 trial. This was opened here yesterday. In terms of factors affecting later progression as well as later strategy, the studies that I listed in the first Excel spreadsheet, which you're probably not seeing right now. Hang on. Are you seeing the Excel spreadsheet or the slides?
The Excel spreadsheet.
Okay, that's good. Also affect later progression. And in terms of later treatment strategy, we have these 4 studies ongoing. We're looking forward to increasing this list. There are a number of variables that are not being studied yet. Immunity duration is one of them. I can tell you anecdotally, in the survey that we're conducting here at LSU, where when monitoring antibodies, we do see them fade after a while, which is consistent with what's in the literature. And I'm going to go back to sharing the slides. These are the 2 studies that Oxner conducted. Seroprevalence within New Orleans, meaning Jefferson and Orleans Parish, looking at both shedding virus by PCR and COVID exposed by IgG. The first thing that you can notice here is exposure generally, but not always, correlates with shedding. For example, look at this here. You can see my cursor, I believe. This is high exposure, but actually not a lot of shedding. And you can see that even within the New Orleans metro area, there's a significant geocoded difference in both exposure and shedding. This makes me think that we probably have different strains of virus circulating that have been introduced into the area from different sources. This was recently published, actually not even, it's a preprint, in the Houston area by Baylor, by Methodist. And what they showed by sequencing viruses is that they've got at least 4 or 5 different introduction events, and that viruses with a certain sequence, in particular the D614G mutation, shed much more. We will be in a position to do a study like this in New Orleans in a month or so, because we're setting up this technology as an FDA-approved test in our lab. Also, another study conducted by Oxner, seroprevalence and any infection in Baton Rouge. Now, what's interesting about this slide is you can see pretty clearly that if you have a public-facing job, particularly healthcare, and also if you're single, which probably means you interact with more people, you have a higher chance of having been exposed to COVID-19. Delivery drivers do seem to not have a lot of luck with infectious diseases, which makes sense. This is consistent with our data at LSU. Our seropositivity rate on campus is quite high. It's 20%. We're over 1,400 participants, but we've managed to keep shedding to a very low level by enforcing masks and social distancing. That's what's going on from the standpoint of biomedical informatics, and I will be happy to either take any questions or pass them along to Ron, San, Dan, or Chindo.
Great. Thank you very much, Dr. Miele. I'm afraid we may have to hold questions till the end, just so everybody has time to give their talks. And I think I am up next. For those of you who don't know me, I'm John Shefflin. I'm Associate Professor of Pediatrics in the section of Infectious Disease, also an Adjunct Professor in Internal Medicine in the section of Infectious Disease. And I am going to share my screen and talk to you quickly about one study that we've completed and 2 studies that we are hoping to start very soon. So the first study I'm going to talk about was a study led by our 3rd-year pediatric infectious disease fellow, Monica Dietrich. And obviously you can see this took a village to get this accomplished. There are a lot of people here who were some significant heavy lifters on this project, especially Betsy Norton and Kevin Swasderick, and certainly James Robinson as well. But this looked specifically, we were interested in looking at rates of seroprevalence among children in Louisiana. And what we were able to do was to get unused blood from Children's Hospital, where I'm sitting now. You know, when patients are hospitalized or come to the clinic to have blood drawn, there's usually some extra blood left over that the hospital holds for about 7 days. After that 7th day, we were able to acquire that excess blood through an IRB-approved protocol and test it for antibodies against the SARS-CoV-2 virus. So between March 18th and May 15th, we collected just under 1,700 blood samples. We excluded several because they were duplicate studies from the same patient or multiple blood draws from the same subject over several days. But in the end, we had just under 1,600 patients that had that were negative for SARS-CoV-2 IgG. I'm sorry, about 750, and then 62 patients that were positive. And if we look at this table here, you can see that overall our spread between male and female was even, although there was a trend towards positive patients being more likely to be female. It was not significant. The median age was essentially the same. So the interesting thing is that when we looked at race, as we've seen in other studies, Black patients were significantly more likely to be positive. This was sort of early on in the outbreak, so even though there were people who identified as Hispanic, there was an increase in their chance of being positive. This is still a relatively low number compared to what we're currently seeing during the outbreak in the state of Louisiana. The other interesting thing is when we looked by zip code at median income, the positive patients had a significantly lower median household income, indicating that these positive patients or patients who with the evidence of prior SARS-CoV-2 infection were coming from zip codes with a lower median household income. This is a map, obviously, of the state of Louisiana. The parishes coded in red are parishes where we had positive samples from. Parishes coded in blue are where we had samples, but they were negative. And then the white parishes are parishes where we did not have any samples at all. So remember, this is again early on in the outbreak, but you can see that there's clearly a cluster in southeast Louisiana, and then in the southwest, close to the Texas border, as well as up in Rapides Parish in the Alexandria area. Looking at over time, the seropositivity increased between March 18th and April 10th, which was early on in the outbreak. And then it increased to about 7.3 to 7.5% in April and May. The graph on the right-hand side of your screen looks at the number of COVID cases in different parishes, again, over time, looking at the when the stay-at-home order was put in place, and then the phase 1 reopening. And again, you can see that there's an increasing trend as expected in Orleans and Jefferson Parish, where most of the activity was going on at that time compared to the other parishes. And this is just another way of looking at our data. So the majority of our positive samples came from Orleans Parish, followed by Jefferson and St. Tammany. Again, this isn't a big surprise based on what we know about the epidemiology of the outbreak at that particular moment. So I'm going to move on now to the first of 2 studies that we are getting ready to launch. The first is a study funded by the CDC. The top title there is the, in my opinion, rather boring title that was on the CDC application, but we really refer to this as the COVID-19 Community Research Partnership. As I said, this is funded by the Centers for Disease Control and Prevention. Our local PI is Richard Oberhelman at the School of Public Health and Tropical Medicine. I'm one of the co-investigators along with Joe Keating and Josh Yukish, who are also at the School of Public Health in the Department of Tropical Medicine, and Patty Kissinger in the Department of Epidemiology. So the goal of this research partnership is to do a a population-based surveillance to look at risk factors of infection, transmission dynamics, because we're doing it over 2 years to look at seasonal and geographic patterns of transmission. And hopefully, we'll also be able to identify correlations between immune markers and reinfection, if reinfection occurs. So how are we going to do this? There are 2 main components to this study. The first is a population-based community serologic survey in the greater New Orleans area. We're going to be sending out mailers, inviting people to participate. They'll fill out a knowledge, attitudes, and practice questionnaire, and then they'll complete a one-time serologic testing. Not very different from what the Oxner study did previously. This study, we feel, is different because it's, for one thing, being done at a later time point, but also because we're recruiting by mail, we'll be able to target a diversity of zip codes, which Oxner did in a different way, but we're going to implement different strategies to get a good representative sample. And then the second, probably the more data-rich portion of the study, is to use healthcare provider networks to perform a serological cohort. This will allow us, by using patients who visit a specific access primary care from a specific healthcare network, to do syndromic surveillance. We're going to do serologic testing on a subset, and also not only get health information from the participants, but also pull data from their healthcare network EMR. So how does this work? First of all, let me say that this is not a New Orleans only study. This study is led by Wake Forest University in North Carolina, as well as a research group called VisNova. And we have partners not only in North Carolina, but also Maryland, Virginia, West Virginia, and Mississippi. So up here, you can see that patients will be recruited. We'll be able to get data from their healthcare network. If they agree, we'll be able to get the data. And then also they'll participate in a monitoring system, not very different from what at least those of us at Tulane, I'm guessing LSU, do as well with the daily symptom check. However, we're going to try to avoid symptom check fatigue by doing it a little less frequently. A subset of them will get a at-home test kit to what I think will likely be collect a blood spot that will be then sent in for IgG testing. If they develop symptoms, then we will send them an at-home test kit or refer them to a test center within their network, and we'll be able to pull all that healthcare data. So this is, I'm sorry, it's a little grainy, but this is some data from, or at least an image of what we hope to be data from the entire network that we're participating in. This is real data from North Carolina. So they've already started. Wake Forest has about 20,000 people already enrolled in this study through a wide diversity of counties within North Carolina. And you can see the red and purple are counties that have the highest seroprevalence of IgG in their communities, but also the fastest growing number of COVID cases as well. So the second study, and I apologize for going so quickly, is one that was just recently funded through the CARES Act and the National Cancer Institute. This is what we're calling the Tulane University COVID-19 Antibody and Immunity Network. It's part of the National Cancer Institute's SeroNet effort to understand what being IgG means, and also to fast track development of diagnostic platforms that can help us understand the function and development, evolution, and memory of the IgG response to the SARS-CoV-2 virus. This effort is led by Dr. James Robinson, one of my fellow pediatric infectious disease physicians. And also on this are Darlene Fusco, who's leading our clinical core, and Elizabeth Norton, who is leading one of the projects, and I'm head of the admin core. So what this study is going to do is we're going to take advantage of existing studies that are ongoing already through at Tulane, as well as some of our partners. This includes the CVID Consortium, which is a Center for Viral Systems Biology based out of Scripps, one of our collaborators with Christian Anderson, Darlene Fusco's ClinSeq-SEER cohort of acutely ill and convalescent COVID-19 patients. We've put together a number of other cohorts to participate this, as well as industry partners and ongoing collaborations that we have with the Tulane National Primate Research Center. In addition to the cores that I've mentioned and these specific cohorts, we're going to have an assay core that's going to focus on development and validation of different assays, including functional assays and neutralization assays. This core will also be responsible for training others within our own center, but also within the entire Sero network, of training other people to use the assays that we've developed and validated. And then we have two projects. One will look specifically at characterizing convalescent serum, and the second will be looking at B and T cell memory. And I saw that Sam Landry is listening in. He has a significant role in the second project here too. So how is this gonna work? As I said, we have a collection of patient cohorts, including a pediatric asthma cohort that we're going to follow them for symptoms. This whole study is supported by the National Cancer Institute, so we have multiple cohorts of cancer patients, both at UMC and Tulane. From those cohorts, we'll be collecting data and samples, and that will then flow down to different functional tests and analyses that we hope to perform. And I'm going to stop here so that we can get back to Dr. Josh Denson, who is an assistant professor of medicine in the section of pulmonary and critical care and environmental medicine at Tulane, and a fine collaborator that I've been working with on other projects as well at Tulane, and a very hardworking critical care doctor who's been taking care of all these patients. Thanks, Josh.
Thanks, John. Let me share my screen real quick, hopefully. All right. Everybody, you can see that, John? Everybody can see that? All right, great. So thanks for having me to speak. Again, my name is Josh Denson at Tulane University. I'm pulmonary and critical care specialist. Today, I'm talking about severe COVID-19 specifically, and kind of addressing different patient outcomes aspects and risk factors. And I know this is our first kind of session, so we'll kind of go through a little quickly. Hopefully I don't run over. Let me know if I am. I have no disclosures today. A couple objectives. I wanted to first start with a few observations about severe COVID-19 and how it may differ from overall COVID-19. The development of some research questions that I have in general, and that I think that this community can help to try to address as we've been discussing. A little bit on research in progress and what we've done so far and next steps. Quickly to I wanted to start by delineating kind of what is severe COVID-19 and how does that differ from the overall syndrome itself. Most patients with severe COVID-19 have one of three symptoms: fever, cough, and shortness of breath, or if not all of them. They have a number of different laboratory abnormalities I won't go into. But most patients, if they're severe enough to be hospitalized, and again, this gets into a little bit, and I'll talk more about it. What is the definition of severe COVID-19? People typically develop a whole host of different symptoms, but to be in the hospital, usually you have developed pneumonia and a need for oxygen requirement, and the patients that are the most severe usually progress to full out respiratory failure. Here's an X-ray showing an example of this, and what we call acute respiratory distress syndrome, or ARDS. These patients have many complications that can be seen in both in the hospital, ranging from kidney disease to blood clots to blood pressure issues to stroke and heart problems. And it's funny, this is a slide talking about ARDS, and before this pandemic, one of the only silver linings is that I don't really have to explain to people what I do anymore, and people aren't asking if I'm an ER doctor or not. And one of the conditions I care for a lot is ARDS, even before we had COVID-19. And now I think everyone on this talk already knows what this is by now, but it was first reported in Denver, where I did my fellowship training. And it's been gone through different sets of definitions, most recently in 2012. And the bottom line is, if you have ARDS, you've had an acute bilateral chest infiltrate pattern pneumonia that has to have some clinical acumen associated with how to diagnose it. And you have to have an oxygen requirement that's not just oxygen, but you have to have some level of ventilatory support. So you have to have some pressure, positive pressure, which is demonstrated right here for the definition. So this slide's a little bit busy. I took an excerpt from the Oxner New England Journal paper that we've talked about a little bit, and I reviewed a lot of the data just to kind of go over the epidemiology of severe COVID-19 and the different outcomes that are being reported in the literature. I'll start with the simplest definition: is what rate are patients hospitalized? So in New Orleans, according to some of our data, and what I was able to review and our references cited below, if you tested positive for COVID back in March and April, you had a 40% chance of needing to be hospitalized, which is pretty high. And if you look further, what percentage of these patients develop acute hypoxic respiratory failure, meaning you didn't just have pneumonia, but you actually needed enough oxygen and were sick enough that somebody labeled you as having respiratory failure? Even if you just tested positive, 10% of cases that tested develop this syndrome, and 25 to 40% of hospitalized cases actually develop respiratory failure. Now, other outcomes that other people have looked at is the need for ICU or ARDS, like I mentioned. And again, the numbers of overall cases follow along with the respiratory failure. So usually if you're in the ICU, if you're developing respiratory failure, or if you develop ARDS, with COVID-19, these things are all going to run together. And you may or may not need mechanical ventilation. But as you can see, if you're tested positive for COVID-19 back in March, April, or even now in this is a conglomerate of all the different data, 10% chance of requiring severe illness. Severe meaning not just in the hospital, but in the ICU or on a ventilator or developing severe pneumonia. And if you're hospitalized, you have anywhere from a 32 to 45% chance of ending up in the ICU. Similar for ARDS, up to about 30%. And then actually ending up on a ventilator. According to different data sources and large data sets so far, you have a 19 to 38%. So about a third of people end up intubated or on a ventilator. And if you're in the ICU, you have about a 70 to 80% chance that you have ended up on the ventilator. And so these are pretty astounding numbers. I know we already know this, but sometimes it's just nice to take a step back and look at the actual numbers and realize the chances of what could happen. Some studies have looked at other patient outcomes, such as shock or the need for vasopressors. Vasopressors are a type of life support that we offer to keep someone's blood pressure up so they don't die. And about 50% of cases in the ICU require vasopressors, and 15% of hospitalized patients. And one commonality between all of these different outcomes is that they can be somewhat subjective. Some of them are not, but depending on the reason why one of these was defined, you can have variable numbers. However, death is one hard outcome that we all know, and that's why it's talked about a lot. You can't really fake it. You can't adjust it so much. And looking at the mortality rates, it's pretty interesting, actually, even though we've been following this for months. Right now, if you test positive, you have a 3 to 9% chance of dying just by testing positive, which I think is very high, right? In the next slide, I'll get a little bit to case fatality, which is similar to mortality rate. But if you're in the hospital, you have anywhere from 17 to 24% chance of dying. So 1 in 4 patients in the hospital are dying from COVID-19. And then about a third to almost half of our ICU cases are still dying from this. And so these numbers are pretty scary. And this is taken from the Johns Hopkins website yesterday. Looking at the U.S., our case fatality rate, which is a mortality rate, number of deaths over number of cases at risk, is 2.9%. And if you guys still remember back in the beginning, we talked so much about the flu, and the flu is at 0.1% at its greatest, right? So it's still a very severe disease. It ranges depending on the numbers that you look at, but just something to keep in your mind. And so I was asked to talk about outcomes and risk factors. And so now thinking about the beginning of New Orleans risk factors. And, when we had the first cases rolling in, they tend to have a phenotype. They usually were sick for about a week, and then they really went over a cliff. And these patients all tended to have a similar picture. They had obesity and diabetes, or they had high blood pressure and kidney disease. And so the question I asked at the time is this just typical New Orleans, right? I mean, most of New Orleans has one of these diagnoses probably, right? So, if you look back at the data actually from early January in China, there's actually a signal there that suggests it's not the case. And we already know this now, as a number of other publications have shown. Looking at that New England Journal paper, if you look at the presence of a primary composite endpoint, which is a combination of ICU, use of mechanical ventilation, or death, again, these different outcomes I talked about, you can see the rate of diabetes among these patients was 27%, compared to 6% if you didn't meet this endpoint. Same thing for hypertension. And as a clinician in the ICU and a lung doctor, when I think of a respiratory virus, the people that I think are most at risk for dying or having a severe outcome are going to be those with COPD, or in this case, 10%, right? And so this is a little unusual, the fact that diabetes was such a strong predictor of how someone, or maybe not a predictor, but had such a strong association with how someone might do poorly. And this has been shown in other studies at the same time. This is a review of early review of the fatalities from 2 hospitals in China. Again, same thing. Diabetes and hypertension are the most common risk factors here. And as we already know and have probably talked about, obesity is a very common risk factor for COVID-19, but both for risk factor and as well as outcome. And different people have shown this, from our group at Tulane, to my old colleagues at NYU, to my old co-chief resident in Seattle, to just different areas. And so this is a clear association. So this led me to kind of develop some, sorry, I think I lost my window here. I'm not sure how, let me see here, 1 second.
No, you're good. We can see you.
Oh, okay. I guess I can't see you guys. All right. So the research question is to know what risk factors and what's the pathophysiology? Can we detect this early on? Are there biomarkers to look for? And what treatment strategies, like we talked about that kind of pathway that we showed earlier, when can we target the treatment? Is there an optimal timing for this? And how can we do that? So, I had this idea in the beginning, right? A lot of these people are, they have 1 or 2 of these diagnoses, or they don't exactly fit into a bucket. And so I was wondering, is metabolic syndrome the commonality that's linking these different unusual diagnoses for a respiratory viral infection? I'd done some work previously at National Jewish on obesity and metabolic syndrome and asthma, and this is what I'm aware of the innate immune system that's related to that. And so for those of you that don't know, but I think a lot of people on the call here is that metabolic syndrome is usually loosely identified by 3 of the following 5 conditions: obesity, glucose intolerance, hypertension, high triglycerides, and a low HDL. And so that was the idea going into it. Is this the unifying diagnosis that increases the risk? And that's what I'm going to talk about today, which was published this past month. So it was a retrospective observational study. We took into account hospitalized COVID patients at the peak of the pandemic, right around that March-April time period. This is from patients from Tulane and University Medical Center. And what we did was we identified patients with metabolic syndrome using a modified WHO criteria. And there's different types of criteria to identify metabolic syndrome. The reason we chose the WHO modified was so that to increase the reproducibility. This is allow you to detect metabolic syndrome using different data sets. We looked at a host of different outcomes with our primary outcome as hospital mortality, and performed several different analyses, multivariate analyses, taking into account age, sex, race, ethnicity, hospital site, and the Charlson Comorbidity Index, which is a validated comorbidity score that's correlated to mortality. We also did separate models for each metabolic syndrome subgroup condition. So we looked to see, is obesity the risk factor alone when you adjust for these variants? Is glucose intolerance the same thing? And then we looked at a host of inflammatory markers that were previously studied in COVID-19, as well as metabolic syndrome. This is our table 1, and I just want to talk. We had 287 patients here. Some interesting things about this is that it was a female predominant population. This is, again, also seen in the oxner population. Very heavily African American, also similar in the oxner population. Again, our patient population is obese. Our group differed from the oxner cohort in that our Charlson comorbidity score was much higher. So it seemed to be a sicker population that we're seeing downtown, perhaps. And then the rates of comorbidities are just, I don't want to say astounding, but they're very, they're very high. 80% of people had high blood pressure, 65% were obese, 53% had diabetes. And what's interesting is we're talking about, again, a respiratory virus, and our rates of COPD are as low as 10%, asthma 10%. So there's definitely something about these conditions which seem to be increased in these patients. ARDS, as I talked about earlier, about almost 30% of people had ARDS. 37% developed the need for invasive mechanical ventilation as IMV. Length of stays are great, and 20% of people died in our cohort. We did subdivide it between ICU and ward just for review at baseline characteristics, but this again wasn't this is how a lot of people have divided people up. But we really wanted to look at, as a cohort study, to try to determine the effect of the association with metabolic syndrome, which is presented here. So if you divide people by metabolic syndrome, again, a very strong, fairly strong female predominance, although not statistically so. And again, more African American. Of course, these patients had a higher BMI, but also had a higher Charlson index score. And when you look at the metabolic syndrome comorbidities, which just to prove that we did separate them along these lines, the 5 different comorbidities are pretty high for these groups. But one thing that's important to note out is even though the control group or the non-metabolic syndrome group, although they didn't meet the criteria for metabolic syndrome, meaning having 3 of the 5 conditions, a lot of them still had a lot of the sub-diagnoses. So a lot of them still were obese or had prediabetes or had hypertension. And so the fact that we'll come to the outcome soon, but it's just remembering that the control group is not a purely non-metabolic cohort here. CRP and LDH were statistically significantly greater for the metabolic syndrome, which is not surprising. And then when we looked at the outcomes, we found some pretty interesting findings. It kind of goes along with the hypothesis here, in that hospital mortality was 25% for the metabolic syndrome patients compared to 10% in those without metabolic syndrome. This had a risk difference of 15% in adjusted odds ratio after taking into account age, sex, and other comorbidities of 3.42. Again, this trended in the same direction for ICU requirement, ARDS, and mechanical ventilation, such that if you have metabolic syndrome and are admitted to the hospital with COVID during this time, 56% of those patients ended up in the ICU, and about half of them ended up intubated, compared to much lower numbers if you did not have metabolic syndrome. Length of stay was longer, and readmissions were a little bit longer, although not statistically different. So this is a forest plot that looks at metabolic syndrome as well as the 5 different subtypes. The adjusted odds ratio is right here in the middle. The 4 different outcomes of ICU, ARDS, mechanical ventilation, and hospital mortality are here on the left. And then here's your confidence interval and estimated odds ratio. You can see the metabolic syndrome for all 4 of these outcomes is strongly statistically significant with a high adjusted odds ratio. Interestingly, though, if you look at at each unique subdiagnosis, for example, obesity, if you look at patients that were obese compared to not obese and adjust for age, sex, comorbidities, race, and hospital site, they were statistically significant at a statistically significant increased risk to be in the ICU, to develop ARDS, and to develop mechanical ventilation, but not mortality. And the same thing kind of held true for pre-diabetes. So pre-diabetes was measured as an A1C of 5.7 or greater, or a history of diabetes. and they had a greater risk of requiring the ICU as well as requiring mechanical ventilation, but not necessarily ARDS or mortality. I think that's kind of interesting. We talk a lot about diabetes, but this took into account even an A1C group that's lower. And so overall, basically it seems that metabolic syndrome may be somewhat exponential. Now, this was not a large enough study to really determine that, but it does seem like it may be an exponential risk factor here. This is from the supplemental data. There's a lot of other data I could cover, but just to show the fact that we looked at 3 different markers, which have been looked at before, and included them each in again, a multivariate analysis using different cutoff values that were published in the literature to have a higher severity in COVID-19. And so if you have, for example, a LDH level greater than 365, that you had an increased risk not only for mortality, but ventilation, ARDS, and ICU. And this held true for LDH and CRP, which is in line with some other studies, particularly this one by Jan. Again, it's just interesting that as potential biomarkers for severity of illness. And this is, again, adjusting for comorbidities, adjusting for age, and taking that into account. So conclusion, so during the peak of the pandemic here in New Orleans, if you had metabolic syndrome, you had roughly 4 times greater odds of severe and fatal COVID-19 outcomes. And these estimations took into account age. So it didn't matter if you were old or young, you still had these greater odds. And it also doesn't take away all the subdiagnoses, so obesity, high blood pressure, diabetes, or prediabetes. But when you put them together as metabolic syndrome, it may be somewhat exponential. And this is ongoing, and people obviously are looking at it. How many studies did we just talk about that are looking at something similar to work to figure this out? And this is data from the CDC just published a week and a half ago, and basically looked at COVID-19 in catchment areas compared to the risk of being hospitalized. And again, I looked at outcomes before, after hospitalization. These are looking at your risk for being hospitalized, and lo and behold, hypertension, diabetes, and obesity again are kind of the main risk factors. And I find it so strange because I would be normally thinking COPD for someone with the flu or another respiratory virus as like a high risk group to be hospitalized, but that's not who gets sick from this disease. And that's really an interesting finding. So why is that? That's something we don't know and we're looking into. Obviously a common pathway might be leptin or macrophage accumulation in adipose tissue, but we're looking at a lot of different things. To finish up here, I think I'm running out of time, there's 2 other next steps is a study we're doing, a larger one, and I'm the PI for the virus COVID-19 registry. It's another similar COVID registry that has, as of yesterday, 27,000 individual COVID-19 admissions from around the world. There is the option to do ancillary studies, and we have a couple in the works. If anyone else wants to, feel free to get in touch with me. And then we have I'm part of another couple studies. I have one goal that's one project looking at biomarkers and metabolic syndrome and severity of disease in COVID. And then I'm part of Dr. Fusco's ClinSeq study, which is a CDC-sponsored study looking at all kinds of different aspects of biospecimen and clinical data related to COVID-19. A lot of people to thank from the Tulane team, LA CaTS, obviously my kids and Tulane and everyone for supporting me. If you want to get in touch with me for questions afterwards, I don't know if we have any time necessarily, but feel free to email me or reach out to me on Twitter. Thanks so much.
Great, thank you, Josh. Yeah, we're a tiny bit over time. I'm sure if there are any burning questions for Dr. Denson or Dr. Miele, we can take them real quick. Either just put them in the chat box or raise a hand or just let us know. Or you can certainly call us, contact us individually if you want to get home and enjoy the beautiful weather we have now.
Yeah, it's a rarity, huh? Sometimes.
Great presentations.
I can't tell if somebody's trying to talk, but I think.
Sorry. I was trying to talk, but I was muted, and then some background noise. So Joshua, that's a great presentation. I have one quick comment. If you consider the endothelial cells as your primary target, as opposed to, yes, of course, it's a respiratory virus, but it's really a systemic endotheliopathy. I mean, the autopsy series that's happened here in New Orleans has shown clearly that there's endothelial damage in every organ that has capillaries. So that may explain why metabolic syndrome increases so much the risk for poor outcomes. It's really nice work. Congratulations.
Yeah, thank you. I think that's what I find so interesting, and I'm, and I want to figure this out because it seems almost like a cardiovascular disease. And John can comment on this, but from an infectious disease standpoint, that's a very unusual thing to have. When we have any viral infection, it happens. Obviously, we have the hemorrhagic fevers, but we don't usually have so many of the viral complications, at least with the typical coronavirus of infections that we see normally. So I thought that's what made me think about all of this, and it makes sense, the endotheliopathy.
Well, that's a good point, Josh. And one of the things that has come to my mind as I cross back and forth between the adult and pediatric world, and you just reminded me of, is that we in the pediatric world have been worried a lot and seen a number of these cases of MISC, or multi-system inflammatory syndrome in children. And when I go see the sickest adult patients, they look very similar.
Yeah, it's funny you mentioned that. There was a case from LSU published on an adult case with MISC. It was a patient of mine who I had at UMC, and it sounded very similar. She was like, I think, in her 30s or something like that. But again, the same comorbidities. And I think that this comorbidity problem trickles down into the pediatric population because we have an obesity epidemic at the same time as this. It's almost like God's playing, He knows how to get us back,
Great. Well, thanks to both of you. I think this is a great talk.
John, you don't have to answer now, but I was intrigued that one of the endpoints of one of the studies you measured is determining T and B cell memory. I would be very interested in finding out a little more detail on how exactly that's done, because I think that's a variable that we all have to pay attention to.
Great. Well, this is one of those things where it takes a village, so it's a large center grant. But I think I'd be more than happy to get you connected with the right people doing those studies.
Thank you.
One short question, Josh, relative to these severe cases, are the neurological findings as common with the loss of taste and smell more prominent in these severe cases?
The which findings?
The neurological findings, loss of taste and smell.
Yeah, that's something you mentioned because everyone knows someone who's got it somewhere at some point. I haven't noticed that per se, and I was actually thinking that I wonder if it tends to be a bit more mild. At least that's some of the literature I want to say I read. The most common symptoms for the severe ones are obviously anything related to the chest. So shortness of breath, a fever, and cough were the most common ones by far. I think the next one on the list was actually GI upset or diarrhea, but the anosmia and things like that have not, at least in my experience in review of the data, that have panned out to be associated with severe disease. I think they're associated with more mild, but I'm not going to take the chance of, if given that. I wouldn't, put my money on it, I guess. Oh, sorry, I was sharing the screen the whole time. Sorry about that.
All right. Well, thank you both very much. I think this is a great time to end this session. And thanks everybody for joining, and let us know if you have any follow-up questions.
Thank you guys so much. This is Megan Bronson. I greatly appreciate all the efforts and all the speakers today. I just want to remind everyone that our next session is scheduled for 2 weeks from today, October 15th. Hopefully everyone can join us again then. And it looks like a last minute question came through the chat. So we'll reach out to you guys via email and see who wants to answer that one. But thank you so much, everyone. We really appreciate all of your time and effort. Have a great night.
Great.
You
Thank
too.
you.
Thank you.